So although mass spectrophotometry has facilitated in the sequencing of the proteins, but there are some problems in that process. So when the mass spectrophotometer, it breaks the small fragments into further small fragments, then the problem is that despite having the small fragments, some molecules that are attached with the molecules, they removed. We will see in the later that when the small molecules that are present with the small peptides or with the amino acids, they are removed, then the mass that is calculated through the mass spectrophotometer is different as the mass of the actual amino acids or peptides. So if we consider that A1, A2 up to A20 is the set of amino acids, and each with the molecular mass that we will calculate later, MAI, have molecular mass, and a peptide P that we know that we break the proteins, then the peptides they are generated, and if a peptide P is equal to P1 and it ends to Pn as the sequence of amino acids, with parent mass MP that we will calculate is the summation of the MP1, and we can denote the partial N-terminal peptide P1 to Pi of mass Mi, because we have to break the protein into the peptides. So this is the N-terminal peptide, and it has a mass of summation of the J is equal to 1 and V is MPJ, like this one. So in this case, as Pi is the partial C-terminal peptide, so it will be Pi plus 1 up to the Pn of mass MP, and in this Pi bar is for 1 is less than equal to I and I is less than equal to N. And we know that the mass spectra obtained by the tandem mass spectrophotometry consists predominantly of partial N-terminal peptides and C-terminal peptides. And we know that every protein is a linear chain. For example, this is protein and is in the form of linear and is connected by a peptide bonds. These are peptide bonds between the amino acids. The peptide bonds start with the nitrogen and ends with the carbon. We can see start with nitrogen, ends with carbon. Therefore, every protein begins with the unstarted peptide bond that begins with N, and another unfinished peptide bonds that ends with C. An N-terminal peptide is a fragment of protein that includes the leftmost, that is also called, because N is called N-terminal, and a fragment of protein that includes the rightmost is called the C-terminal. So we can see that this is the general structure of the protein, having the amino acid and they are connected by the peptide bonds. So a mass spectrophotometer, as we know that it typically breaks the peptides into further smaller fragments, and we call them as the P1, P2 up to the Pn at different peptide bonds, and detect the masses of the resulting partial N-terminal and C-terminal peptides. For example, if we have this peptide GPFNA, it represents different amino acids present in the peptide. So the peptide may be broken into the N-terminal peptides, because we know this is the N-terminal and this is the C-terminal. So if we consider that it is broken from the N-terminal, then the first peptide will be, smaller fragment will be G, second will be GP, then GPF, GPFN. And if we consider that it is broken from the C-terminal, then these peptides will be PFNA, then the FNA, then the NA, and then A. So moreover, while breaking GPFNA into GP and FN, as I have told you that what's the problem in detecting the sequencing of this protein by using the mass spectrophotometer. So if we break it into GP and FNA, it may lose some small parts of GP and FNA. and resulting in fragments of a lower masses. And for example, we know that the peptide GP, this one, may lose a water molecule. So if a water molecule is removed from this one, the mass of the GP will be reduced. And similarly, the peptide FNA might lose an molecule of ammonia from its peptide. And again, if ammonia is removed, then the peptide mass will be reduced. So the resulting masses detected by the spectrometer will be equal to the mass of GP minus the mass of water. So we can calculate the mass of GP, and we have to minus the mass of water, and we know that the formula of water is H2O, and we know the mass of hydrogen is 1, molecules they are 2, and we know the mass of oxygen 16. So 16 plus 2 is equal to 18. So we have to minus the value of water from this HP. And similarly, because the molecule of ammonia is removed from the FNA, then we have to remove the mass of ammonia from this. And we know that the mass of the nitrogen is 14 plus hydrogen atom 1, 3 atoms, so 14 plus 3 is equal to 17. So we have to remove the 17 from this FNA peptide. So peptides missing water and ammonia are two different ion types that can consider in fragmenting a peptide in a mass spectrometer.